The goal of the Human Genome Project. The primary goal is to identify and catalog the approximately this many number of genes are there in the human DNA. We know very well these many number of genes are very, very important to control the various characters. So, what are all the characters controlling what all the genes are present in what all the chromosomes? If you are understanding, then manipulation of the genes is very easy for us. Why? Because accordingly we can control the genetic disorders, right? So I already told you in a couple, the girl is having one of the family genetic disorder history. The boy is having one of the family genetic disorder history. So both the genetic history, being a genetic counselor in the developed country, we will be counseling the particular couple. If this is the case of genetic history, then the probability of getting the genetic disorder we can understand, right? Now we are coming further. Next major phase, technology transfer and ethical consideration and project coordination. Here, many industries, came for establishing the research work with the Human Genome Project organized by 13 countries. Okay, in this, ethical issues are kept in the place, in the sense the dead human beings and the old human beings from their body, the DNA is taken. Like this, you should not calculate. We should calculate. They took the DNA from all the human of various age, and then they evaluated for understanding the gene expression differences. Right? The Wellcome Trust. Okay, and as I already told you, and U.S. Department of Energy and other countries together they included this project. Right? In the human biology, at the genetic level, healthcare, agriculture, energy production, and environmental remediation. Okay, so all these things they taken for consideration. Why? Because they need to improvise not only the human digital characters, not only to bring a great variety in the plant breed, they thought of bringing many more advancement by making a superior breed. By making a superior breed. So even genome used two major approaches. He expressed the sequence tag and sequencing the entire gene. What do you mean by express the sequence tag or EST? Expressive sequence tag means those specific DNA segments can make the RNA. Those specific RNA can make the protein. Sequencing the entire genome. Sequencing the entire genome means the sense the entire gene content of an organism is called genome. That genome are made up of many alleles. Okay, and those alleles in which it is getting expressed. That they sort of understanding. Okay, here DNA from cell are isolated, fragmented into small pieces. So the hidden sentence of the second sentence is why they need to fragment the gene by using what enzyme? By restriction. Okay, they got a fragment. With that fragment, what they're going to do? They're going to multiply the fragments. Those fragments are nothing but what? Alleles. Responsible for what? For a character expression. So the character expression alleles, we brought in many copies with the help of the plasmid vectors. Or else we can say the extra circular DNA of bacteria, extra circular DNA of the yeast are very important for duplicating the genes into many copies. Frederick Sanger is a great scientist who did the DNA sequencing technique. See, if you're talking about uh, Sanger method is used for many important sequencing process. See, for example, if you're talking about Frederick Sanger dideoxy method for DNA sequence. If you're saying Sanger's method for protein sequence, we should be very careful. Sanger's dideoxy method for DNA sequence. Sanger's method, if you're saying plainly, Sanger's method, not dideoxy, is not protein sequence. So we should be very careful with all this set of sequencing technique. YOX stands for East Artificial Chromosome. BAC stands for Bacillus Artificial Chromosome. What is this chromosome denotes? Cytoplasmic DNA. That means it not indicates nuclear DNA? Yes. What is the cytoplasmic DNA? Extra chromosomal circular double-stranded DNA. Extra chromosomal circular double-stranded DNA which is nothing but back in the yolk, which is very important for the given gene to duplicate into multiple copies, right? Now we are coming to the second set of explanation. In the salient feature of the Human Genome Project, the Human Genome contains 3164.7 million base pairs. Okay, here the total length of the human genome is this many number of base pairs, 3164.7 million base pairs, right? So in these base pairs, everything are not coding for a character. Everything are not coding for a character. Only certain amount and that too negligible amount, not huge amount is coding, not huge amount. Only certain amount is coding for the character. Okay. The average length ranges from 3000 base pair, but sizes differs greatly with the large known human gene dystrophin at 2.4 million bases. 2.4 million bases, right? So a typical gene consists of 3,000 bases. 3,000 bases. So if you're talking about the gene size, is dystrophin. Dystrophin is the biggest gene in the human being. Okay, and this dystrophin gene codes for the muscle protein, right? There is a message you need to understand. Now we are entering the concept. If you're talking about the salient feature of human genome, the total number of gene which is present in a species is 3,000, much more than the 1,40,000 genes. So the all okay here, if you're talking about all the human beings are having 99.9% similar type of genes. Only 0.1% allele differences are there. For example, nobody is having crocodile nose. One human being is having crocodile nose. The other human being is having elephant nose. Other human being is having parrot nose. It's not like that. So everybody, 99.9% of the human being are having the same type of nose shape. Only the difference nose size and structure at the micro level, right? That is because of the micro change the nucleotides of the alleles, right? Alleles are the gene copies, we all know very well, right? So now, if you're talking about the functions of more than 50% of the genes remains unknown. Still, we not understood. Okay, this is what is going on. Okay, this allele performs what, that allele performs what, this gene copies performs what, we don't know, right? Now we are coming to less than 2% of the genome codes are protein. The majority of the human genome, over 98, 90%, 90% does not directly code for protein. Okay? Only 2% codes for protein. That is the reason why, before the commencement of the particular subtopic session, I told you only negligible amount of alleles are coding for a character. Most of the alleles are not coding for any character like this. We already narrated you indirectly. The repeated sequence makes a large portion of the human genome. The repeated sequence is a hidden sentence. I may repeat sequence. I made up of a number of repetitive nucleotide sequence. I won't do anything. I'll remain as a numb. Okay, my existence is waste. Like this, repetitive sequence are same. But if you're going to the high technology, for example, in CBSE, they don't repetitive sequence or junk DNA. Its existence is worthless. They kept it out. But if you're going to the advanced version at research level, when you are going for master of engineering or PhD, at the time you will understand these repetitive sequences are very important. If it is not there, that particular coding sequence will be broken very easily. The coding sequence are completely shielded by repetitive sequence. Coding sequence are completely shielded by repetitive sequence. If the repetitive sequence are not removed, okay, if the repetitive sequence are not removed, then the coding sequence will not get matured. That is required. But repetitive sequence protects the mature DNA from the defective enzymes. So that is why what the repetitive sequence are completely uh, blocking the or completely masking the or completely protecting the coding sequence. The repetitive sequence not having coding sequence, not code for anything, but protects the coding sequence. Dot. Now we are coming for repetitive sequence. How it works exactly. It is not going to bring any uh, ancestral characters for any generation, right? It is just a protective structure. Now, the chromosome 1 has the some genes, a 2969 and Y has the fuse 231. The human genome is contributed to the 23 pair of chromosome. right? The chromosome 1 carries the highest number of genes among them. Okay, here Y chromosomes has fewest gene. The fewest gene, the Y chromosome. The chromosome 1 is the maximum gene. So, which is the maximum gene, which is the less number of gene that we already told you, right? So, the Y chromosome indicates only the male chromosome, which is present only in the case of male sex, right? So, scientists have identified 1.4 million locations where a single base pair difference occurs in them. Here, you know very well, the process of finding chromosome location for the disease associated sequence and tracing human history. Uh, Here, disease associated sequence. See, for example, some newborn babies that enter the world with genetic disorders like Alzheimer's disease, Parkinson's disease. There is not the issue of the parent, there is not the issue of the mother. So, because of the gene combination, the zygote area, because of the defective gene inheritance while making the gamete. So, this arises randomly and brings a defective genetic character. So, that should be identified. Okay, before going for the marriage, that should be identified. Okay, to the particular couple with a proper narration indicated. Why? Because they will be having this level of probability of getting the genetic disorders. Like this, we need to narrate for, right? Single nucleotide polymorphism, single base difference in the DNA. If there is a difference in a single point, obviously many issues will come to the DNA as well as RNA and the protein, right? That is the way you are comparing. Now we are coming for the second phase explanation. Here, application future challenges. Before going to that particular place, we'll go for related videos. Please listen to that particular video animations. You can go to university portal. Now we are going for example. One examples discussion, hot question discussion. Explain the historical context that leads to the initiation of the Human Genome Project and its significance. Human Genome Project launched in 1980 as a response to the rapid advancement of the genetic engineering, right? To understand the gene makeup of an organism. To understand the gene makeup of an organism, right? So, uh, if we're talking about the Human Genome Project, will help to understand the gene makeup of a human being, like this we told you, right? So, gene makeup of a human being means character governing alleles are present where exactly we can understand. And why this human being is very tall, why he is very short, why he is tall, why he is having tan skin, why he is having the white skin. So, all these information we can understand if you know the proper nucleotic sequence of an DNA, right? Now we are coming to the next point is describe the Sanchez sequencing technique. This Sanchez dideoxy method is exclusively for understanding the DNA sequencing method. This DNA sequencing method will help us to understand what nucleotide is there in the first question, what nucleotide is there in the second question, what nucleotide is there in the third question. Like this, we can understand by doing the Sanchez sequencing technique, right? So DDNTPs, the expansion of DDNTP is dideoxynucleotide triphosphate. So these DDNTPs will stop the DNA synthesis process. By using DDNTPs, we are understanding how the DNA is organized in terms of nucleotide sequence. The nucleotide sequence, if you are understanding, then controlling the nucleotides in expression is very easy for us, right? Now we are coming for discuss the ethical, legal, and social issues associated with the genomic research and how the Human Genome Project addressed them. You know very well, ethical, legal, social issues includes privacy, genetic and discrimination, and informed consent. Okay, here, uh, this much amount of gene manipulation can be executed. This many number of gene manipulation can be executed. That much amount of gene manipulation we can execute. To a certain level, we don't go. Like this, what chemical we are using for gene manipulation. Everything should be discussed with the ethical committee of the government body of the respective country before doing this. Okay, because some gene manipulation is possible and we can bring the good character. Some gene manipulation possibility is not there. Even though we are doing some side effects will come, everything should be discussed with the ethical committee and documented. After that, we can go for the Human Genome Project analysis, right? Now we are coming further. We discussed about the work examples, tips for the exam. Okay. In the tips for the exam, we need to understand the various subtopics, side topics. Then entering into the concept is very, very easy for us, right? So in Human Genome Project, which is the biggest gene, which is the smallest gene. So which gene is having maximum repetitive sequence? What is the advantage of the repetitive sequence? What is the disadvantage of the repetitive sequence? Which gene will go for gene expression? Which gene will not go for gene expression? Everything we should understand, right? Now we are entering into the presentation of the next subtopic. Yeah. So in this particular subtopic, we are understanding the application and challenges. In the application and challenges, what we need to understand is basically here unraveling biological system, interdisciplinary collaboration, okay, uh, deep understanding of the biological system from the molecular level to complex level, right? Unraveling biological system. So the knowledge about the DNA, RNA, and protein and the cell behavior cells, everything should be understood. Okay, in this Human Genome Project, right? In the sense, this allele is responsible for this character is present in this particular location of the chromosome. That we need to understand. Then manipulating the allele, then doing the trial and error method to understand the side effect, and then bringing that particular gene sequence in a particular routine order or bringing a digital variety should be systemized by doing a lot of analysis. Okay, that should be programmed, documented properly, right? Interdisciplinary collaboration. Okay, experts in genetics, genomics, bioinformatics, and various biological specialties, right? Here, the collaboration of scientists from the diverse field, both in public and private sectors. Okay, so here the geneticians will say the different information. Okay, that will be kept in a very easy order by bioinformaticians. Okay, so and then what are the probabilities of gene mixing? That will be told by genomics technicians. So all the experts we should come in the same lane to perform this beautiful human genome project so that what the entire thing will be fulfilled. So that what we can avoid the genetic disorder transmission from one generation to another generation. New approaches to the research. What do we know new approaches to the research? The human genome sequence has revolutionized the biological research approaches in the past. Scientists focused on studying individual genes or a small number of genes, right? So, some genes performs very aggressively, some genes will not perform very aggressively, some genes will express when there is a requirement, but if there is no requirement, the gene will not perform very nicely. So the gene's performance level at a different time period depends upon the amount of cell requirement, the amount of organ requirement. For example, I'll say to you here, in the menopause woman, in the post menopause woman, there is no sufficient enzyme production for the egg production. Okay, the egg production will not happen in the post menopause woman, but because enzymes are not there to make the egg. In a male who is 50 plus, the sperm production quality is not that much great. And the sperm number is not that much great in the 50 years old individual. If you're talking about the men who is less than 35, less than 40, will be having very good number of sperm cells. For that, having a very good gene expression, which is expressed properly before 40, but not expressed after 40 years. What is the reason for that? Okay, what is the reason for that? That understanding is very important to know the Human Genome Project operation, right? So now, if we're talking about the system biology, we know very well not only the alleles, not only the protein, not only the RNA, okay, and not only the DNA existence in the cell. Many parameters are involved in controlling the gene expression. Now we are coming to this is the research. Research is identifying this is associated with the genetic variation, right? Okay, here. I already told you not only Parkinson's disease, Alzheimer's disease, okay, here basic disease, like this, so many genetic disorders are present in number, right? Uh, and that genetic disorders, causative element or the allele responsible for this particular disorder should be determined with its chromosome location. So that giving appropriate treatment is uh, programmed well programmed properly with a set of experts, right? Now we are entering into DNA fingerprinting. Before entering into the DNA fingerprinting, let me solve some questions of the Human Genome Project. Human Genome Project is a mega project launched in 1990 with just some important goal. So, what is the primary goal? We need to stop genetic disorder inheritance from one particular era to the other particular era, or one particular generation to other particular generation. Okay, and then we, for example, even in Bhopal issues, what happened is because of the methyl isocyanate gas leakage, many even are giving the defective birth, even now also in Madhya Pradesh. The same issue we are experiencing in the case of Japan, even some babies are coming to the world with the defectiveness. The children are coming to the world with the defectiveness. Why? Because of the genetic disorders arise because of the mutation. You know very well in Japan, Hiroshima and Nagasaki, the World War included the atomic bomb blast. Because of that, many genetic variations happened, and still now also many individuals are coming in the world with disorders. In Japan, in the Madhya Pradesh, India, like this, we can give many examples, right? So that particular genetic disorders, if you're understanding properly, and if you're giving the correct measure and giving the gene therapy, it will be useful. For that, your project is meant for. Why the Human Project we need to do? Okay, energy waste, energy, money, time, okay, huge even muscle work, everything we are giving for what for? I need to get benefited. So how do you get benefited? So allele information, if you know properly, with the correct location in the chromosome, then treatment is very easy for us. In the Human Genome Project. So that is the answer for this, right? Okay. Now we are to the next important question. We discussed one more question. Okay. Now we are entering into the what was the primary role of the Human Genome Project? To clone human gene, to determine the sequence of the entire human genome, to create a genetically modified organism, to create genetic diversity of human mind. Second option, determine the sequence of the human genome. Right? Second option is the correct answer. Okay, my goal is to understand the sequence of the human genome. This place of the human DNA and the reproductive sequence. This place has the coding sequence. Like this, we need to understand. Right? Now we are entering into the next question, number three. In this question number three, how many base pairs are estimated to make the human genome according to the Human Genome Project findings? Of course, we all know even 1 million base pairs. 1 million base pairs. Or 3 billion base pairs. 3 billion base pairs. Right? So, here, uh, this 3 billion base pairs information are kept bioinformatically. That means in terms of database, which is evaluated very easily whenever we require exactly, right? That is the reason why the Human Genome Project is including the usage of bioinformatics tool. Okay, where the total baseline what we found is 3 million baseline. Right. Now we are coming into the third question discussion. What is the significance of the SNP in the context of Human Genome Project? Okay, how many varieties of genetic disorders? Okay, location associated with the genetic disease and tracking the human genetic history for identifying location associated with the genetic disease. Right. Uh, So this is the major information we need to understand. Single nucleotide polymorphism, right? This is associated sequence means nucleotide sequence, right? That is a message we need to understand.